Again, we're going to be looking at regulatory strategies. Later on in the semester, the second half of the semester is when we go much more specific, okay? And we start talking about specific regulations of enzymes. For now, we're going to be just using the strategies and talking about overall strategies. Okay, now the enzyme activity is going to be regulated in 5 principal ways. This is the 5 principal ways of regulation that we're going to talk about. We're not going to get fully into everything here because we just can't. It just is too much. So some of it will only become really important in like chapter 31. Okay. Now, the allosteric control by regulatory molecules, this is probably the one you think of the most, actually, if I was to guess. The alternative enzymes that catalyze these same reactions but exhibit different catalytic and regulatory properties. You've actually seen hints of this so far, but we haven't really talked about much. You know, like how we talked about there's the different myoglobins and such. Okay. The reversible covalent modifications, we've actually talked about this, haven't we? Yeah. And you've gotten that from another class as well. We can discuss that a little bit. The irreversible proteolytic cleavage to yield an active enzyme product. And 5, which is the final one, is controlling the amount of enzyme present. Now, this is a bit more complex and doesn't involve the actual enzyme itself, right? If we're controlling the amount present, that's going to be things that are not having to do with the enzyme itself. Going to be things like making the enzyme, right? And moving the enzyme. And so that's why that shows up later. So the allosteric regulation is going to enable the control of your metabolic pathways. The pyrimidines and purines are going to be the aromatic groups that compose the unique bases found in DNA and RNA. Yes, those are the ones we had talked about in DNA, RNA. Remember the purines and pyrimidines. Those were those bases. The aspartate carbamylase, which is ATP case, is going to catalyze the first step in pyrimidine biosynthesis. This is a committed step. A committed step is going to be the step in the pathway where the products of the reaction are committed to the ultimate synthesis of end products in the pathway. Think of it like this. At a committed step, we are committed to going through this reaction. Okay? You can't stop after a committed step until doing usually several reactions later. Okay? And so this is the one where you have committed to doing that cycle, you have committed to doing that pathway. The typical reason for committed steps is they're going to be irreversible under cellular conditions. They're going to be catalyzed by allosteric enzymes. Mostly committed steps happen because something that would be necessary to go backwards in the reaction is no longer there. Okay? So like if you produce CO2, very likely the CO2 is going to leave, right? And so you're committed if you produce CO2, almost exclusively. Okay, so this right here is the ATC. This is going to catalyze your committed step in the pyrimidine synthesis, this being that one right here. Here's the carbamyl phosphate. Here's the aspartate. There's AT case, or ATC. And this is the N-carbamyl aspartate. Now, everybody know what that means where we have several arrows? That means we don't care about the rest of the reaction. So you have at least 3 steps to get to here. Okay? And for our case, we just don't care, do we? This is the part we're talking about. That's the committed step. Okay. Notice that we have cleaved the phosphate off. Then we're going to do several steps. We're going to get the cytidine triphosphate. This is the CTP. That is the C version of ATP. Okay. So CTP is going to inhibit the AT case. This is actually feedback inhibition. This is a product that we make later on in the reaction that will inhibit the activity of the enzyme. So again, this is inhibition. The exact definition your book gives is inhibition of an enzyme by the end product of a pathway, which we're looking at the specific example that's usually used for this. Okay? Now, this is going to ensure the pathway intermediates are not needlessly formed when the end product is abundant. Where would you want to target when you're trying to do some sort of inhibition? Okay? So, let's say that we have a staircase that has a problem on it, okay? People are going to fall off and hurt themselves. They go up it past halfway, right? Do we put a sign halfway up the staircase where the problem is? No. Where do we put the sign? The bottom. Why do we put the sign at the bottom? That is your, or another way of saying that would be your committed step. Literally step, right? I know. It's a pun and an example. Okay? So same thing here. This is why feedback inhibitions typically target certain steps. They're committed steps. Okay? And so you target those to inhibit this, because if you inhibit after this, you're going to waste stuff, aren't you? Up to this point, all of these reactions can go backwards. At this point, you can no longer go backwards. You have to finish this. Okay? And that's why there's no backwards arrows. Can everybody see that there's no backwards arrows there? Yeah. Because this would be your committed step. After this, you're going through. Right? Like if you do the first few steps on a staircase, you're committed until you get to the end, aren't you? All right. Now, this is again that rate of formation here. This is versus the CTP concentration. And as you can see, what happens with our rate of formation, our rate of this reaction as we increase our CTP? It gets slower, doesn't it? Okay? That's not the direct product, is it? No, what are we looking at? We're looking at feedback inhibition, aren't we? As we make more CTP and have more CTP around, this is going to inhibit that formation of that the intermediates. That first step here. Okay. Another way of saying this is it's like a staircase that's getting full, and you go, oh, that's full. We should probably not go up the staircase, right? Now, the CTP, this is going to be an allosteric inhibitor. So the CTP is structurally different than the reaction substrates. That does not look anything like these, right? Everybody agrees on that? Nobody thinks they look alike? Not at all, right? And so this is not binding to the active site, is it? No way. It's completely different size, completely different everything, right? And so this has to be allosteric interaction, and meaning that is an allosteric inhibitor. The allosteric or regulatory sites are going to be sites that are distinct from the active site at which the substrate binds. They're instead going to be sites that are at a different spot that will bind to different groups. The CTP is going to be an allosteric inhibitor. This is going to exert its effects by binding at that distinct allosteric site on the ATC ACE. And again, that is not the binding site, not the substrate binding site, right? Not the active site. Okay, and so many allosteric regulated enzymes do not follow the Michaelis-Menten kinetics because, again, they're going to have where you make the product, you go a few more steps down, you make that product, and then it has inhibition, right? And so by definition, because they're making product, they're going to diverge from Michaelis-Menten kinetics, aren't they? Which, anytime you have inhibition, you diverge from Michaelis-Menten kinetics, right? Okay. So again, this is what that looks like. Notice that we have diverged from that very nice logarithmic pattern, and instead now have a more sigmoidal pattern. Everybody see it okay? Like our previous examples of our inhibition. Okay. Again, that sigmoidal curves, if you forgot, result in the cooperation between the subunits. And this means we're having structural changes for that inhibition. Okay? Okay, so we have the ATP case. This is going to consist of separable catalytic and regulatory subunits. The native AT case is going to consist of the 6 catalytic, which are C for catalytic, and 6 regulatory, which are for regulatory subunits. This means that the full complex is going to give you the C6R6. It'll be organized into 2 catalytic trimers and 3 regulatory dimers. And that's that specific orientation that we had talked about. Remember how in like hemoglobin we had 2 dimers, right? Okay, so the isolated subunits will combine rapidly when mixed. So if you mix the different subunits, they'll actually combine and come together and form your full units on their own, spontaneously, and they'll do it rapidly. So the p-hydroxy mercure benzoate is going to react with our crucial cysteine residues and our ATC. Is everybody okay with me saying ATC instead of ATCase? Okay, fantastic. Okay, so the treatment of the ATC with the substance right there, the p-hydroxy mercurate benzoate. Notice the mercury right there. Everybody okay with that? Do you guys realize that that's that same group we had talked about in organic chemistry, and we've done some of those mercury reactions? The hydroxy mercury. And then we had other things. Usually not benzoate. Yeah. Cool, huh? Little Easter egg. So again, this is actually the cysteine right here. We're going to disassociate the enzyme by using this substance here. What's going to happen is we'll get the catalytic subunits, which consist of 3 chains. So we'll have a C3 that comes off. We'll also have a regulatory subunit that'll consist of 2 chains, which will give us our R2. And this happens actually because of this reaction right here. Now, the allosteric interactions in the ATC are going to be mediated by large changes in your quaternary structure. 2 catalytic trimers are going to be stacked upon one another. So you have 2 catalytic trimers stacked. Everybody envisioning that in your head? Kinda? Okay, so there's going to be significant contact will only exist between the catalytic and regulatory subunits. Zinc ion is going to be critical for this interaction of your R chain with your C chain. This is actually what it looks like in a top view. This is the side view, by the way. Can everybody see your catalytic trimer here? And this is using your regulatory dimer. Okay. And then notice that this is all 6 that you can see of your regulatory dimers. That's the R6 right there. 3 sets of dimer, right? Okay. What are you not seeing from the top view, by the way? We only have 3 trimers, right? We only have 3 catalytic. The other 3 are hidden under them because they're stacked. Okay. And so that's why you need the side view, by the way. This right here is the R chain that you're seeing right there. This is that zinc right there. This is the C chain right here, which is going to form that catalytic trimer right there. Okay. The Pella, this is going to be a bisubstrate analog that's going to represent an intermediate. This will be along the ATC catalytic pathway. And so again, it's going to be an analog of one of these substances along that pathway. So the Pella, this is the name of Pella. It's not really that important, and I think we've actually seen this before, if I remember correctly. This right here is then going to be the bound substrate. That would be your reaction intermediate. This right here is Pella, that is the analog of that. Can everybody see how it's the analog? The big difference being this is a charged nitrogen here, this is a neutral one here, right? And instead of oxygen here, it has a carbon. So that's a more stable, less energetic. Okay? So the active sites of the ATC, Pella is going to be a potent competitive inhibitor of your ATC. It's going to bind to and block your active sites. It'll bind at the boundaries between the pairs of the C chains within a catalytic trimer. Each catalytic trimer is going to contribute 3 active sites to the enzyme. Each of them has an active site on that catalytic C chain. Okay. This is actually what that looks like. Remember, pala is an analog of the substrate that's a bit more stable and binds a bit more stably. Okay? And so what we're seeing here is this is the catalytic trimer. Can everybody see that okay? Okay. These are then the palas within them. And see how they bind at sort of the edges of this? Everybody see that okay? Okay. And then this is actually where it binds in. Notice, by the way, it does it at the edge again. And so these bottom ones here are green. These top ones are yellow. What are they? Why? Why are these yellow and why are these green? This is yellow, isn't it? This one is green. And so this is binding here, isn't it? Yeah. This is this one. Everybody say it okay? Yeah. And that's why that's the edge, and it actually binds between them, with 2 of them being involved in each binding. Okay? Okay, so the ATC, this is going to exist in 2 conformations. This is why this is going to have a regulatory effect with these R dimers. It's going to help change that conformations, right? And notice we'll have a T and R state, just like we talked about for things like hemoglobin, right? Okay, and so the binding area palo is going to cause a structural changes. This is going to convert the compact, less active, tense state into the expanded, more active, relaxed state. The R state will be the more relaxed, more active state. R state referring to this gap here, it being more released, right? T state, tensed, gap has closed. Okay? Less active state, which one is this? More active state, right? Okay. What induces that conformational change? Binding of the pala, right? Okay. Which is an analog of what? Is an analog of an inhibitor. Of the substrate, right? Yeah, it's an analog of actually the reactive intermediate of the substrate, to be specific. Okay? So essentially it would be the in-between of here. So if you were to look at this reaction, you would have the activation energies and intermediates between these, right? And it's going to be like that. Okay? Is that making more sense? Mm-hmm.
So the pala and then the hydroxy mercer enzyme, they both bind to the ATC, correct?
Okay, they're different things, but yes, they both bind to ATC. Yeah. These split the 2 chains apart. So we have the 2 trimers. This is going to help split those. And we'll be able to fragment these things so we can get the pieces and look at them.
They both do stop the ATC.
Okay, so this actually is going to dissociate the enzyme, meaning you're going to take it apart. Yeah. So that's not actually an inhibition like we're talking about here. This is just more of a structural part where we've taken it apart to see the different parts and explore them. Okay. On the other hand, this one right here, pala, is a intermediate. So this is an analog of a substrate. This will bind to the binding site. And this is where we're exploring what it looks like when it binds and what it does. Okay? We need to use a more stable analog because otherwise it would be too damn fast. And we can't actually explore what it looks like. Okay? And so in this case, what we're doing is we're seeing that it has a cooperative behavior, right? That the substrate actually is going to make it more active. Okay? And so more substrate means they'll be more active, right? Okay. Okay, or another way of saying that is the substrate will help to activate it. And we're using Pella in place of the substrate. Because otherwise the substrate would go to product, and we don't want that. We don't want to look at it, right? ATC, but this binds at the active site. Okay? And there's 3 active sites for each of the trimers. Okay, and so the ATC, this is going to exist in your equilibrium between the T and R states. That's that equilibrium like we had talked about before. The allosteric coefficient is actually going to be related to the ratio of the concentration of the enzyme in the T state to that of the R state. Or another way of representing that is that formula right there. So our allosteric coefficient is going to equal the T over R state, right? Okay, what does that mean in this case? So the R state is the what state? More active or less active? More active, exactly. The T state is the less active state. I'm getting you to use that terminology because oftentimes these will not be fully turned off or fully turned on. Okay? You'll have more active, less active. So in this case right here, if you have a very high allosteric coefficient, what does that mean for the activity of this? It's very low, right? Yeah. If you have a very low number, you'll have more in the R, right? Which means it's more active. Okay? Okay, so your T state, this is going to have a low affinity for your substrates. It has a low catalytic activity. It's favored by the absence of substrate. If you have no substrate coming in, then you don't really want to do this reaction and take away what substrate happens to be around, right? The R state has a higher affinity for the substrate, is more active form, and is favored upon binding of the substrate. So if you bind one of the substrates, it's going to favor binding another one, okay? And the more substrate you have, the more active it's going to get. Okay? Activity. It does. Yeah, so, no. Pella is not an inhibitor. So, Pella is actually an intermediate, not an inhibitor. Okay, so let's be very, very clear, okay? So yes, Pella will act as a strong inhibitor, competitive inhibitor. It will increase the binding and activity of it. But the reason it acts as an inhibitor is because it's a stable intermediate, and you can't go forward with the reaction. So when you bind the Pella, it blocks that binding site, okay? But, I'm sorry.
It keeps it from binding to the substrate.
Yes, when it's bound to that site, it would keep it from binding the substrate. But you do have a weird behavior that if you added the correct amount of Pella with the normal substrates, you could actually have more activity, okay? Because it will make the overall enzyme more likely to bind and more active. Does that make sense? Better sense? Okay. 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 Okay, we got that. All right, so this is again cooperativity and the homotropic effects. You may remember cooperativity not from enzymes, but the hemoglobin, right? Which is binding but does not have enzymatic activity, right? Well, technically it does, but we don't want that enzymatic activity. So the cooperativity, this is going to be the property by which the subunits will cooperate with one another. And you notice that this actually has a very similar sort of thing to hemoglobin, doesn't it? We had an R state and T state there. When we bind one site, we get more activity on the others to bind further, right? It's just in the case of hemoglobin, it's not an enzyme, okay? So we're still sort of learning the same things, just in different ways, right? Now, the homotropic effects is going to be the cooperative effects of substrates on allosteric enzymes. So this is going to act on enzyme active sites. It's going to generate our sigmoidal curve again, because we won't have perfect Michaelis-Menten kinetics anymore, will we? We're going to have feedback. Okay. This will impact the ATCase activity. It'll do it by altering that T to R state. Okay. Again, in this case, though, this is not going to be due to the inhibition in this case, that cooperative activity. It'll actually work to activate it, right? Okay. But yes, Pella is the inhibitor normally. Okay. All right, so limiting modes for cooperativity. The ACCase is going to adhere to the concerted all or none mechanism for allosteric enzymes. So your entire enzyme is being converted to T into R. You won't get a partial T, partial R, right? And we discussed two different ways of looking at hemoglobin before. This is that one that you convert the whole thing. Okay. Most of the other allosteric enzymes are actually going to exhibit features of both the concerted and sequential model. And you may remember those models from previous chapter. Does everybody kind of remember? Yeah. Okay. Sequential means it partially transforms as you go along. Concerted means it's either one or the other. Okay. This is unusual. The ATCase is unusual. Most of these other allosteric ones do actually do a little bit of both, like the hemoglobin did. This one's all or none, concerted, that's it. The sigmoidal curve for the ATCase is going to be a composition of two Michaelis-Menten curves, like all sigmoidal curves actually are. Okay? There are composition between different Michaelis-Menten curves if you were to solve for it. It's just that sometimes they're not just two, sometimes it's more complex. But in this case, we have two curves. We have the curve for the R state, and we have the curve for the T state, right? Because again, this is concerted, you only get the two curves that are possible. Here's the curve for the R state. And you can see that this would be a Michaelis-Menten curve, isn't it? It's a logarithmic curve. Very nice. And you can actually see that the T state's the one being followed here, and then we're getting R state here, aren't we? Okay? Can everybody see that? How they sort of match one spot and then we match the other? Yeah. And again, that's because of that cooperativity, right? As we're increasing the aspartate, we're making it so it favors what state? R state. That's what you're seeing here. Neat, huh? Okay. So again, the allosteric enzymes are going to display threshold effects. That's what we're seeing. It's called threshold effects. The transition is going to occur within a narrow range of substrate concentrations, as we can see up here, right? It's only a very small range that starts happening. As composed to the rest of it, pretty much just stays the same after that, right? And so this is actually zoomed into this front curve where we can see that better, right here. And the threshold effect is going to be the activity of the allosteric enzymes is more sensitive to changes in the substrate when we're near the Km than the Michaelis-Menten enzymes with the same Vmax. So right here we actually see this quite nicely. And so we can see here as we come across, this is that difference right here, right? And this is the range where we're following this behavior first. And then as we get to the larger range, we start following the other behavior, don't we? Okay. Can everybody see it okay? Fantastic. So the allosteric regulation is going to modulate the T to R equilibrium. ACCase is going to be in the T state when bound to the CTP. Now, what was the CTP again? That wasn't the substrate, was it? And that wasn't the Pella either, was it? No. That was the feedback inhibitor, right? And so this is a final product some steps down. It's coming through back in. It's binding at a different site, not the substrate site, right? It's binding at the allosteric site. And then it is causing the T state to be favored. And so we're getting an equilibrium shift towards the T state, which does not favor binding to the substrate, right? Which in fact is non-favorable to binding to the substrate, not favorable to calic activity, right? Okay. So the CTP binding site is going to exist in each regulatory chain in a domain that does not interact with the catalytic subunit. This is what it's going to look like right here. So again, here's the catalytic subunits. This is a side view. So this would be a trimer set right here on top, trimer set on the bottom, right? And then here we have the R chains. Those are that 3 dimers. We can only see 2 of them here, okay? And so what we're seeing here is that if you look here at the ends here, they have sites where you can bind the CTP. Can everybody see those CTP binding over here? Notice by the way the loops, did you guys see those loops? And we'll talk more about those loops, okay? But we gotta get through certain materials first. All right. Anyways, can everybody see that binding change? So we stabilize the T state. It can no longer open that gap to get to the R state now. Okay. Okay. We'll start up on here next time. You have a wonderful day, and I will see you all Thursday. Okay. Sounds good.